Good evening, everyone. I am Utkarsh, and it's my pleasure to welcome you all to the sixth day of our lecture series on the topic Antibody Drug Conjugates, the anti-cancer magic bullets. Before we begin, I would like to remind you that if you have any questions, please submit them through the chat box or reserve them until the end of the presentation. It is now my honor to invite Mr. Eswaran to deliver today's lecture. Thank you.
What a delight, what a joy to talk to a group online. And this afternoon into the evening lecture is entitled Antibody Drug Conjugates, the Anti-Cancer Magic Bullets. Strictly speaking, the first word in the title should be slightly expanded, not just antibody, but monoclonal antibody. So you will see sometime down the road in this lecture, things ending, materials or compounds or drugs ending with MAB, monoclonal antibodies. This is the recent subject relatively. These monoclonal antibodies, I don't want to announce myself as a student of either biochemistry or nanotechnology or biology. We do it in a collaborative manner with our collaborators who are trained in these subjects. But it is known that these can be derived from white blood cells and are very specific, target specific. Drug can be a range of drugs, in this case, drugs which are anti-cancer drugs. And you join the two of them. So the abbreviation, one more abbreviation, ADC, Antibody Drug Conjugates. Again, at the beginning, I have to warn my audience, this is not a popular talk. I have been giving many popular talks to young students, college students, school students, and general public. This is not a purely popular talk. We are all students, serious students of science, and this talk is meant, like other talks, to highlight the work I have done in the last 10 years, especially my retirement days since 2012, which is 12 years plus, under very challenging conditions. Where challenging conditions bring the best out of man. And mark these words of mine, because half a century later, you will be also addressing some student groups, and you will remember what I said to you. Monoclonal antibodies, I defined them to the extent that I could. I defined ADC, antibody drug conjugates. Why should we talk about this? What is the need for antibody drug conjugates? The antibody, the monoclonal antibody, which can be very specific, target specific. That means it will only attack that particular cancer cell and no other cell. The drug, both are good, but both have limitations. They have terrible side effects. Somewhere I bring into this title another small chemistry which led to the Nobel Prize in Chemistry in 2022, which will be discussed partly but not in detail. It has come to be known as click chemistry. You bring two things together, they just click. Obviously, energetics is involved. At the same time, how do we synthesize ADCs? How do we characterize them? And what is DAR, drug antibody ratio, drug antibody ratio? What is a good value for DAR? Is it a value of 7, between 7 and 10? Is it a value above 10, which is not useful? And what is the process by which we synthesize this ourselves? And that is two drugs I'm going to mention. One has been mentioned before in a previous lecture. This trastuzumab. I told you MAB means this is a monoclonal antibody. There is something very specific about trastuzumab which I want to mention in the middle of this lecture. Doxorubicin is an anti-cancer drug very commonly used in the hospitals. but it has terrible side effects. Who does not know the side effects of chemotherapy? If there is somebody suffering it in a family, the whole family gets destroyed, not only because of the suffering, but also the financial constraint. Was this or these drugs mentioned in the recent central budget, union budget of the central government in 2024? These are some of the topics we are going to cover during this evening's talk. So it's a very specialized talk. I will make it as simple as possible. So at the end, any one of you wishes to raise questions, I'll be very happy to answer. Here is an example, a three-dimensional picture of an antibody drug conjugate as imagined. It's a schematic for ADC. It's a 21st century magic bullet for treatment of cancer. These are attractive as to the primary pharmaceutical industry, as these are site-specific in nature. Yet, these are biological missiles for cancer therapy. Somebody at the end of the lecture, a word I will not use, but I'll just mention, theranostics. Then you can ask me that question at the end of the lecture. At the same time, these are useful in tracking cellular phenomena and enzyme functioning, imaging, and biomarkers. So on the left-hand side, there is this Y-shaped thing shown in different colors. That is the antibody. And there is a cytotoxic drug shown. That drug is doxorubicin in our case. And these two are combined together by a linker. That linker is the aromatic azide that we have prepared. And in this case, unlike the reactions which led to Vishwamyine formation or fluorazirman preparation or synthesis, the azide N3 group is intact in these reactions. So the nitrogen is not lost. This is a reaction where nitrogen is retained. This is a brief schematic of what are antibody drug conjugates. I explained the two of them work better than either of the two. You see, the side effects of many anti-cancer drugs is cardiotoxicity, the case of doxorubicin, hair loss, nausea, vomiting, and what all. But these new drugs which I'm introducing today, the ADC or the antibody drug conjugate, where the drug is conjugated through a linker molecule. The toxin is the drug linked through that small bond shown, which is our molecule, the so-called cross-linker. And in this case, as others have done, they work mostly on homo-bifunctional cross-linkers with two identical groups on either end. Ours are an example of what is called a hetero-bifunctional cross-linker. That means the two end groups are dissimilar. One could be an N-hydroxysuccinimide group, easily replaced by an amino group in a nucleophilic substitution. The other azide group can be made to undergo a reaction which we have not discussed in the strict sense, where nitrogen is not lost. This is the basic idea and the need for these conjugates. I wish to emphasize on the right-hand side, I introduce a reaction. You see, the advantage of going periodically to universities abroad. I haven't gone to Japan, I have not gone to China, but the western part of the world, I've gone periodically. You hear things which are current, au courant. Somebody speaks better French, I will understand and hear from the person. What is in vogue, what is in fashion, what is going to catch the attention of the world. The reaction on the right-hand side is the so-called click reaction. What is being demonstrated is a triple bond, an alkyne, combining with an azide, an aryl azide or an alkyl azide, where the nitrogen of the azide, three nitrogens are retained, and the triple bond binds to this, what was previously known as 1,3 dipolar cycloaddition reaction. This was discovered in the 1960s, late 1960s, by Rolf Huisgen in Munich, who should have become a Nobel Prize winner. I do not know whether he lived long enough to be named as a Nobel laureate, because the Nobel Prize came in 2022. I had written to him as early as 1973, and he asked Professor T.R. Sheshadri, Fellow of the Royal Society, London, to write to him about me. He wrote and gave me a position in his lab, but I did not join his lab because I wanted to do more biochemistry. So I did not want to do organic chemistry, which I understand was my feeling at that time. How right I was, you will learn from me in personal discussions. So I preferred to join the student of Professor Adolf Buchanan, Nobel laureate in biochemistry and medicine and so on. So he done such tremendous work as a chemist, biochemist, bordering on medicine. So I joined the lab, Max Planck Institute for Biochemistry in Martin Street, very newly set up and very modern. Today, this Martin Street is no longer a village. It is a center of science and internationally recognized. So it's a very highly specific reaction. When Rolf Huisgen did this work, it took 5 days or 6 days to complete the reaction. Then came two people. The name of one is Meldal, who was almost overlooked for the Nobel Prize, and Sharpless, who had already won a Nobel Prize previously, shared the second Nobel Prize of his, along with one more person whom I will mention. I can mention the name now. Maybe in one of the next lectures, we'll see a small video from this lady by name Caroline Bertozzi. It is known as Bertozzi reaction, a gift to biology. Bertozzi reaction, gift to biology. Click reaction is considered and soon was considered after 2001 to be very important for the drug industry. This turned out to be true largely. So 2022 came the recognition as a chemistry Nobel Prize to Meldal, Sharpless, and you can see that it happens so smoothly. You just click the two units together. There was an azide on the right-hand side. There was a triple bond on the other side. The two of them joined together to give you, can you start the video again? Yeah, I'm sorry. The azide is on the left-hand side and the alkyne is on the right-hand side. When you brought them together, what Sharpless did was he catalyzed this reaction by adding a little copper. I think was it copper 1? And then he added a little ascorbic acid. This made the reaction go very quickly, but it still took time. There are advantages for this reaction, which will be shown in the possibly next slide or so. So click chemistry is useful in many areas, as you see here in the green is mentioned, if you can read, high chemical yields, high chemical yields. To that right, you have again the figures below that is the fact that it is highly regiospecific and stereospecific. Highly regiospecific and stereospecific. You get only 1,4-disubstituted compounds. Can be done in many solvents. At the same time, it is not affected by the presence of oxygen or by water. And if you notice, there is a large thermodynamic advantage in doing this reaction. So the reaction goes very fine, the forward direction. And we have to also see that there is a distinct exothermic relationship between the reactants. This has been referred to by somebody as spring-loaded. The reagents are spring-loaded. That means the click reaction I learned when I was in Germany, somewhere around, I think, was it 2006. Then we came back. I came back. I started doing click reactions in our lab. We published a paper in 2011, a very elaborate paper. But I did not know of the other two varieties which I learned in the year 2016. This was classical copper catalyzed click reaction, cycloaddition reaction. The second one that came up is a strain-promoted alkyne azide coupling or cycloaddition, SPAAC, SPAAC, strain-promoted alkyne or azide alkyne cycloaddition. The third one is a very highly efficient reaction. The ways of doing this is the reaction goes very smoothly. But more importantly, there is yet another variety which may not be called click or strictly speaking, a Diels-Alder reaction. Diels-Alder reaction, I used to call it Diels-Alder. My students called it Diels-Alder reaction. It's a very important reaction for the examination for most undergraduate courses and many, many entrance examinations, because 1928, this reaction was discovered by Diels and Alder, who never met each other. I do not know whether they met each other in the Nobel Academy when they got the award in 1928. But what is interesting is there is yet another way of doing this reaction. That reaction is the one that I'm going to mention that just now, inverse electron demand Diels-Alder reaction, IEDDA. But remember, the second Nobel Prize after 1928 came to Hoffman and Woodward and Hoffman. But I think by that time, Woodward had left this world, so he was not named in the Nobel Prize. And they don't generally name more than 3 people in the prize as people who share the Nobel Prize that year. Inverse electron demand Diels-Alder reaction gives 100% yields instantly. Unfortunately, I have not yet graduated to doing this particular variety. Click reaction, we published a detailed paper in 2011, which you can read on the internet. If you don't get it, ask us, we'll send you. SPAAC is a topic we are going to touch this evening. Strain-promoted alkyne azide coupling, which does not need the presence of copper or any ascorbic acid. This is a very nice way of doing this. I go back to the mention of Huisgen. I think he lived into his late 90s, is still very active at that age. In many countries, people, scientists who are very devoted, live very long and continue to contribute. See, there are 2 possibilities. The addition could have led to a 1,2 isomer, or it can lead to a 1,4 isomer. Please believe me, this reaction has been controlled so well that there is no trace of the 1,2 cycloaddition product. The reaction is regiospecific. At the same time, it's highly stereospecific. But that was copper catalyzed. I'm talking about today, not the copper catalyzed click reaction, but another variety which I've said, SPAAC, which is shown in the subsequent slides. So see, all the 3 varieties are shown under the titles A, capital B, capital C. You have now become familiar with the classical click reaction. You become very familiar with the classical click reaction, but it is catalyzed by the presence of copper and ascorbic acid. The second example under title B is a reaction between an azide and an alkyne. But note, what kind of an alkyne? A very special alkyne. I never thought in my undergraduate or postgraduate days that I will ever hear about or purchase or make a trans-cyclooctane. Octane, this is an octane, not an octane, octane. That means that you know how triple bonds are. Triple bonds, the bonds which are shown on the left of the triple bond and the right are shown in a bent manner. That is untrue. It must be at 180 degrees with respect to each other if the hybridization of the 2 carbons is sp hybridized. It's not sp3, it's not sp2, it's sp hybridized. So the shape shown here is totally wrong. Therefore, the cyclooctane, substituted cyclooctane, must be under too much of strain. So it will like to release the strain very quickly by undergoing a very wonderful reaction, the same click reaction with the N3 group, the azide group, and give the same substituted 1, 2, 3 triazoles, but fused to the cyclooctene ring, which are because the triple bond has become a double bond. It is an example of a Diels order reaction, something I never knew about till 2016. At that time, I had shifted from St. Stephen's College to the Regional Center for Biotechnology, Faridabad. There, they did not do so much chemistry as biology, molecular biology, structural biology, and so on. So I was among the few people who's doing hardcore organic chemistry. But we did not do the experimental part. We wrote a review article, which was published in a very well-known journal, IUBMB in 2000, I think 17. 2 interns had come to me, one from Amity University, Noida, and the other from Bharatiya University, Tamil Nadu. They stayed with me for 6 months and wrote that review article with me. Under the title C is a tetrazine on the left-hand side. And on the right-hand side is a trans-cyclooctane. Now, trans-cyclooctane, just like the cyclooctane, must be highly under stress and lot of tension. It will like to open up and become a free saturated system, as you see in the right-hand side as the product. So the cyclooctane, trans-cyclooctane, TCO, trans-cyclooctane, reacts with not an azide. So this is strictly speaking not a click reaction, but it is inverse electron demand Diels-Alder reaction. Whatever we have read about Diels-Alder reaction, where we have a dienophile combining with a diene, we know what is the position of the HOMO and the LUMO. I'm not going to get into the details of this, which will be shown in the next slide, but remember for a moment that the trans-cyclooctane is combining with the tetrazine, forming a 1-2 positioned nitrogens, reduced system, and a gas, nitrogen gas has escaped. The nitrogen has escaped. And there is a cycloaddition, inverse electron demand Diels-Alder reaction. Remember, as I mentioned here at the bottom, again, is mentioned the name of this lady, Caroline Bertozzi. What she did was, click reaction is not just a reaction in the chemistry laboratory. It happens in the human cell with great efficiency. And these reactions are called bioorthogonal. That means the reagent comes in a bioorthogonal fashion, carries out the reaction. It does not interfere with any cellular processes. Moreover, Caroline Bertozzi was very clever. She put a fluorescent label in her case, and she was able to see this reaction happening inside living bodies, mice, what have you, human beings, and she was able to show precisely where the reaction is happening, what are the changes, and she could follow these, especially in a mechanism which we will discuss in one of the later classes or later lectures, Caroline Bertozzi. And I'm sure there are many women members in this group of ours. It's a very uphill task. It has been over the years for women to do science. So we have many examples. One of the examples from our own college, well respected. But even in America, it was very difficult for women to do science. I met a Nobel laureate in 1976, and he happened to be from Australia, but he was living and working in UK, in Britain, and it is somewhat just like the physicist we respect very much, this organic chemist from Australia, then in UK, he was dumb and almost deaf. So his wife, who was also an organic chemist, would accompany him everywhere. This is John W. Conforth, Nobel laureate. So I was talking to him in the ship, and he told me, my wife is more intelligent than me. She came to England before me. But I'm sorry, but being a woman, and we got married and she had kids, and she had to give up her science. But our students from St. Stephen's College are not doing this. Many women students in the last 2 decades or so, or 3 decades, are continuing to do science at the highest level. I can give you a separate lecture on this, which is not the intention today. So as a woman, to do science is very challenging, even in a country like America. Now, I think America is slightly becoming a little more open. We may have a woman of Indian origin may becoming possibly the president of the United States. I'm not speaking for anybody, don't get me wrong. This is just an off-the-cuff remark. So the statement that women find it very difficult. Caroline Bertozzi became the president of the American Chemical Society before she became the Nobel laureate in 2022. I have seen her video, part of which we will show you in one of the subsequent lectures. She's so smartly dressed. She talks so emphatically, with so much of clarity, and has now been recognized with this very important award. Next slide shows you how do we make use of this chemistry, which I mentioned to you, to make antibody drug conjugates. Please remember, antibodies are very costly. The one that I will mention to you is an example made in India. The drug which I'm going to mention to you shortly is made in China. We don't make, please tell me I'm wrong. Check up the internet, subsequently come back to me on the mobile or via email or WhatsApp. If I remember, this particular monoclonal antibody, which I'm going to discuss in the next couple of slides, is made in Bangalore in India. The drug is imported. The one in the middle, if you see the orangish or brownish circle on the right-hand side, a Y-shaped antibody. In the middle is a cross-linker, not made by us, but this is an example of a homo-bifunctional cross-linker, as both the ends of the molecule have an N-hydroxysuccinimide, which can be easily displaced nucleophilically by an amino group, leading to the antibody drug conjugate. In the bottom portion of the slide is shown the use of a heterobifunctional cross-linker. The functional groups are on the left-hand side is an NHS, N-hydroxysuccinimide. On the right-hand side of the molecule in the middle is a very common azide group. And the azide group must undergo a click reaction. That means on the one part, there must be a triple bond. On the other part, there must be an amino group. The amino group on the left-hand side will displace the N-hydroxysuccinimide group easily by incubation overnight, forming an amide bond. On the other hand, on the right-hand side of the molecule in the center, we will have a click reaction. It may take place very, very easily, very smoothly to give a 1,2,3 triazole, 1,4 disubstituted triazole, and the drug molecule and the antibody are covalently linked to each other. I hope you understand. When they are covalently linked, the covalent bonds require a lot of energy to break. Of course, an amide bond can be easily hydrolyzed by either basic conditions or alkaline conditions or acidic conditions found in our body, in different parts of our body. The triazole is not going to break so easily. So this will become eventually a slow release, low dose treatment. And what is the treatment we are thinking of? Treating cancer, including breast cancer, with these drugs. This is the background to our work, which we will introduce to you shortly. But the last few slides will be again a paper from 2017 highlighting the importance of how these studies are carried out. Again, when I get into that part, maybe I'll ask my student, Dr. Sujit Thakur, to say a word or two. I'll ask Manur Bhav Arya to say a word or two, because I don't want to parade myself as a biologist. I'm a chemist. Am I a proud chemist? No, no. I am a hard-working chemist. I know I can make molecules. Then if I made them, I have to prove that I made them. I cannot just say, I made them. I just mix the two. It is not a kind of a, what is it called? Non-covalent interaction. These are characterizable compounds, but these are very high molecular weight. So the techniques you have to use cannot be techniques of chemistry. You have to transcend, go to techniques of biochemistry, and eventually techniques of biology. Do not have any doubt. Many of our students, including your head of the departments, Dr. Vibha Sharma's classmate, is doing so brilliantly in America as a biochemist using the latest modern techniques, and they are making great discoveries, as it were. So you should take courage that it is certainly possible for each one of you to transcend yourself. Now, I'm going to tell you what we have done recently, the last few years. This is an example of a work done by us. On the left-hand side is the monoclonal antibody, which is called by the name trastuzumab. I told you MAB stands for monoclonal antibody. And this is amino groups abundantly. In the middle is a molecule which we bought commercially. It's very costly. It's called NHS-BCN, N-hydroxysuccinimide of a, shall we say, cyclopropyl fused cyclooctane, bicyclononane, as it were, 8 plus 1, 9 carbon atoms. And this is cis, because the two hydrogens are pointed upwards, and there is a triple bond, which is highly strained. So, we, on the right-hand side, the blue circle, you have the drug to which we have an azide group added. That azide group is also prepared in the same manner by displacing a certain reagent, which we will use subsequently, you'll see. But since this is a cyclooctane under great strain, you know, in second year you read, or possibly even the first year you read Bayer's, ring strain theory. He even went, you see, he said 6-membered rings will be very safe. He said 5-membered rings may also be safe. And he said large rings, they will be impossible to make. And he went wrong again, Nobel laureate. And the student won the Nobel Prize before him. 2 years later, the professor won the Nobel Prize. Rolf von Bayer, or Bayer. He went on to say that 3-membered rings, 4-membered rings can never be prepared. One should not go beyond one's brief, should not extend things without logic. Now, you know what we have done? We have taken trastuzumab, we have combined it through a cross-linker NHS-BCN, NHS bicyclononane, where there's a triple bond under strain, and the triple bond has undergone a reaction eventually with the azide and has formed a triazole ring, and the blue portion shows the drug. So this is trastuzumab doxorubicin antibody or monoclonal antibody drug conjugate made by us. Just claiming that I have made it. See, each of the components is very costly. But treating cancer is also a very, very expensive affair. The next slide, the homo-lumo concept in inverse electron demand Diels-Alder reaction or Diels-Alder reaction, IEDDA, is explained. I do not want to take much time because this will be very time consuming to explain this slide. Now, this is work published in 2017. We have been talking to each other for nearly, I think, 40 minutes or 35 minutes. You see what is shown in this schematic. On the left-hand side, there is a tumor cell. Outside, there is a pre-targeting. You can see how many carbon atoms are in that ring. TCO. TCO stands for trans-cyclooctene, not octine, trans-cyclooctene. And what is the reagent just about the arrow? The drug conjugated to what is called a tetrazine. So the tetrazine is to react with the trans-cyclooctene. In the middle is your so-called, antibody. These are to go and hit the tumor cell specifically. So on the right-hand side, the reaction is carried out in vivo. That means inside a living, something like a mice or a rat or what have you, and the compound actually reaches the tumor cell alone and does not touch the, what do you say, any other normal cells, highly specific. And if you see the fact that we have the dark bands on the two diagrams shown below, and that is cell binding, tumor uptake, and these are very considerable. You see the right gray thing is herceptin absent. The left one, which is the dark band or the bar, is herceptin positive. Herceptin is the antibody where this compound is going to bind in an animal or a human being with cancer, especially breast cancer. This is called herceptin positive tumor. And this happens in especially breast cancer. There is another variety of cancer which I mentioned the other day, triple negative breast cancer, where 3 markers are not detected at all till the patient is in a terminal stage of the disease. So this is shown in 2017 that this antibody drug conjugate is very effective in killing or targeting the herceptin receptor. This is published in 2017. Similarly, here you see that this slide, you have the antibody on the left-hand side, and maybe I should stop here for a moment, and we do an IEDDA reaction, and we prepare the antibody drug conjugate, and we characterize it by its mass spectrum. The mass spectrum, the molecular weight, if you see, is 51,652. There's a big band of 24,919. So these are possibly the so-called heavy chain and light chain of the antibody. But what is interesting is the diagram on the right-hand side shows you an SDS page and analysis of these 4 possibilities. I will ask, you see, my friend, my student, now PhD, Dr. Sujit Thakur, to say a word or two. Sujit, आप बोलेंगे ये क्या SDS पेज क्या दिखा है?
Yes, sir.
थोड़ा आवाज कम करके, वॉल्यूम कम करके.
Yes, sir. Here is a SDS page, and in the extreme left there is a molecular marker, and when the antibody is present after the treatment, you can see a continuous band on nearby 60, 50, upper 50. So this band is just cut, trypsin digestion and everything. After that, they got the a specific ESIMS mass spectrum, which is shown in the figure. First is 51,652. Another one is 24,919 and 25,827. So these 2 bands are showing. First is heavy chain, which is generally obtained from 50,000 nearby, and another is light chain, which is generally obtained at nearby 25,000, which is showing that in the antibody there is some drug is attached, which is already picked up after the SDS page gel, which is showing here.
Thank you. This is not my work. This is not our work, but the work Dr. Sujit did with me, and it is included in his PhD thesis, approved by 3 examiners, will also be touched upon again. Next slide shows us. This is again 2017 work from literature. And I do not know whether Dr. Sujit Thakur would want to explain this. Let me make a few sentences here. In the diagram figure called small a, there is a trans-cyclooctene monoclonal antibody. On the left-hand side this is shown how this has been made. And this is then combined with a drug, and the antibody drug conjugate through the cross-linker that we have just now seen, a trans-cyclooctene. That is in figure A, it is an IEDDA experiment done in a rat. Now, see, students of chemistry do work in a test tube or in a flask, but a biologist works differently. They have to work in a living rat in which a tumor has been artificially generated. It is sequential addition of an antibody and then a small molecule. A tetrazine tracer is then administered to the rat, and then eventually ADC, or the antibody drug conjugate, is localized. And you can see this, a radio label was also used. In C, you can see that the actual antibody drug conjugate has gone specifically to the tumor site. This is remarkable. That's why they are called as magic bullets for cancer treatment in the 21st century. But monoclonal antibodies became very well known when President Trump was then the president, suffered from COVID-19. He was treated with a monoclonal antibody. The monoclonal antibody drug became available in India too. One box cost 1,20,000. There are 2, capsules or ampules there. So 2 patients had to share the cost. Still, 1 ampule cost 60,000. And those who could afford 60,000 rupees, those who could afford, went to some of the specialized hospitals and got treated with monoclonal antibody during COVID-19 times as well. But we are talking about cancer or tumor. This tumor in the rat is, generated by what is called, artificial methods. By chemical treatment, the tumor is generated. Then this entire process of treatment with an antibody drug conjugate is carried out, and you can see it live because the molecule is fluorescent. You can even see it under, a microscope of a variety where you can take video shots. Such instruments are available in the institute that I was presently working at that time, not now, the Regional Center for Biotechnology in Faridabad. So you can actually take a video shot of what is happening. Where is the drug going? This is exactly what won Caroline Bertozzi the Nobel Prize. She could precisely show where the click reaction is happening, where the click type reaction is happening. And she could show the fluorescent labels present. And especially, she was working on viral diseases as well. So it was very important and became very important in COVID-19 times. Next slide. We are close to the end of our lecture. How do you characterize antibody drug conjugates? This is nothing to do with the biological work. I will ask Mr. Manubhav Arya, he's a PhD student now and spent about 1.5 years with us, to briefly read out the way we characterize these ADCs. Manubhav, will you do so? Yes.
Yes, sir. Yes, sir. So, as it is mentioned here also, the basic techniques like ESIMS, which was mentioned in the previous slide, and also
stands for electron spray
ionization mass spectrometry. And
every lab
also,
does not have it. Yeah.
yes, sir. And also liquid chromatography, size exclusion chromatography, and reversed phase high performance liquid chromatography. So these are the techniques which are used in characterization of ADC, and also this DAR is another important factor which is involved in characterization of ADC. This, sir referred in the beginning of the presentation. This is antibody drug ratio. Okay, so this is the basic parameters which are required in order to understand an ADC which is developed.
What is the value of DAR that is preferred, Sujit?
Sir, preferred DAR value is 7 to 10. Yes, this is okay. After that, this is considered that antibody drug conjugate will be cytotoxic.
Okay, next slide. Thank you, both of you. Next slide. See, we can skip this slide. The next. This is what we did. I repeat, our cross-linker is shown. It is an azide, aromatic azide, based on 3-hydroxyanthonic acid. The carboxylic group has been converted into an N-hydroxysuccinimide group. That means there are two separate processes. One is the protection of the carboxyl as an NHS group and the generation of an azide group. In step 1, Sujit, please read out in step 1, what are we doing?
Yes,
We
sir.
are taking this cross-linker and
Yes,
combining it with the anti-cancer drug, doxorubicin.
Yeah, in the step 1, we take doxorubicin, make the doxorubicin base and attach with our heterobifunctional cross-linker, that is methoxy cross-linker. Overnight incubation, we get
NHS is displaced. NHS group is displaced.
where
We have an amide bond.
Yes, sir. So here NHS is displaced and make a amide bond. After the bond formation, here is an azide is also available. So this is the part 1. In the second part, we use the antibody and another, that is BCN NHS sister, which is we used here, commercial available. And after this overnight incubation, this antibody is attached with the BCN. So here is a triple bond, a strain triple bond, and here is a azide bond. So in the final
hold on. Next slide. See, this is precisely what we did. This BCN antibody conjugate was combined with the drug conjugate with our cross-linker. And when they were put together, the strain promoted alkyne azide coupling reaction took place very efficiently to give us the drug antibody drug conjugate, which is shown as a large molecule. The color of which is shown on the right-hand side in the tube. It is reddish in color. Our sample of trastuzumab drug doxorubicin conjugate is before you, made in the Regional Center for Biotechnology and then tested further. Next slide. The FTIR spectrum is shown. It's only for purpose of showing it. Next slide. This is a zeta potential size measurement. There are two sizes which Dr. Sujit has mentioned earlier, possibly high, large chain and the small chain.
Yeah.
Both are present in the next slide. The fluorescence spectrum is shown. The circular dichroism, CD is shown. Very, very interesting, yet to be analyzed. Totally, we need some students to analyze this. Next. Biological work was done Dr. Prosijit's lab in Regional Center for Biotechnology. I owe a debt of gratitude to him because its students did this work, and I will leave it to Dr. Sujit to explain it very briefly.
Yes,
the
sir.
A, B, C, D, the 4 parts.
Yes. A is the Western blot, which we are doing with our ADC in the concentration of 10 microgram per ml and 20 microgram per ml. So based on this Western blot, we have seen that the marker which is known for the anti-cancer activity, that is beta acting cytochrome C, caspase 9, cleaved caspase 9 BAX. So these marker is overexpressed, which is taken from and experiment performed densitometry, where we get the specific results that what is the role of each marker. So in the densitometry, there is a Bax beta actin, B is Bax beta actin, C is cleaved caspase 9 or caspase 9, D is cytochrome C. Overall, this densitometry results in showing significant inhibition of by our antibody drug conjugate in anti-cancer cell
And
line, that is 4T1
this is
breast
also
cancer cell line.
efficient cell killing. Apoptosis is cell death.
Yeah.
Efficient apoptosis indicating activity in 4T1 breast cancer cell lines. This is in vitro work. This is not in vivo work. In vivo work, next slide. See, trastuzumab is being made in Biocon in Bangalore. Very costly. Kiran Shah Majumdar or Kiran Majumdar Shah, in 1978, she went to Narayana Murthy, Infosys owner, founder, one of the founders of Infosys, and he was very generous. He gave her 10,000 rupees to start a workplace in a garage. From that garage, with an investment of 10,000 rupees given by Mr. Narayana Murthy, she built a Biocon, whose worth today is 36,600 crore rupees. Her company is listed in New York Stock Exchange. What am I trying to tell you? Science to make money? No. There is another aspect to it which we must note in St. Stephen's College. You are serving society. You are not serving yourself. That man alone is alive. That man alone is alive who lives for others. Others are more dead than alive. St. Stephen's College, one of the mottos is service, love, truth. It's not only that I'm trying to imagine and impress you by saying she has generated so much of money. Ask her, her own husband a couple of years ago, I'm sorry to say, was suffering from this very major disease. I do not know what happened to him. He is that Shaw mentioned in her name, a British national who was her husband. I don't know whether he overcame this dreaded disease. So many people are dying. So many families are destroyed because these are very costly to treat, terrible side effects. So Kiran Shah Majumdar tells young people, don't be a job seeker. Don't be a job seeker. Be a job provider. The trastuzumab came to us from Biocon. The doxorubicin is made in China. It's a very complicated drug. We made the crosslinker. We joined the two of them. Why did we do it? We did not want side effects of either of the two. We did not want that the patient should suffer more than required. The possible side effects be less, low dose, slow release should be a good possibility. So we are almost at the end of the lecture. The previous slide, what did it show, young persons? Previous slide. Yes. You see, this year, the budget put up by our finance minister, Nirmala Sitharaman, 2024-25 budget, she cut the customs duty for a couple of drugs, which are shown in red color. Trastuzumab, deruxtecan, I'm finding it difficult to read it, osmiantinib, and durvalumab. Two of them are MAB ending. They are monoclonal antibodies. The other two are possibly derivatives of the first one that is mentioned, trastuzumab. That means the government of India announced reduced customs duty. Why? Because to make them more affordable to our patients. But more importantly, that you must understand, when I say customs duty was reduced, that these are coming from abroad. So our patients are taking things coming from abroad. Question of customs duty comes only when you import. Can we not produce these things in a college like St. Stephen's College, Hindu College, Miranda House, or Zakir Hussain College, where from some students are present? I would like, towards the end of this talk, ask a question. What is in store in the future? We have published one paper in 2012. I said it's 2011. I corrected. It's 2012 in Monash, FTFO Chemistry. And we have another paper under review. And the third one is under review as well. And Dr. Prasanjit has to yet to say yes, because they are going to add work on mice, possibly give us confocal microscopy video. So you can see where our drugs are, our antibody drug conjugate is going and binding to which part of the tumor and so on. And you can visualize the binding. Towards the end, I want to say this in the nanocarbon lecture, polarized Irman, single walled carbon nanotube, and reduced graphene oxide. I did not mention, if you use near infrared laser, you can do both. What is it? Diagnosis and treatment in one go. That means if you shine light laser, near infrared laser, where the nanocarbon has gone into the tumor or cancer cell or whatever, therefore, and the nanocarbon will explode as if it is Diwali, Crackers, Diwali crackers for that matter. Diwali crackers inside a cancer cell, the cancer cell dies. You can be so specific. You can see it happening. You can actually hear it happening. Many of you are too young to have ever undergone an echo diagram and echo recording of your heart. You can actually hear loudly the beating of your heart. Similarly, we can actually see and hear the killing of a cancer cell. This is described under a new word. I will announce the word. This is called theranostics. Therapy, treatment, combined with diagnostics. For what purpose? Patient has to go first get his diagnosis done, then he has to go to another place, or her, himself, that lady has, or a gent has to go to another place to get treatment. This entire process is very, very psychologically damaging. Can we finish everything in one go, diagnose and treat simultaneously? This looks like a dream, but there are journals, a journal called Journal of Theranostics. So many, many papers are being published. Are we through with my presentation, young people? Can you tell me if that is so? Is there any other slide? Yeah, have you included this slide? Oh, in 1976, this is in my grandpa's Nobel Tales. I am on the ship with Nobel laureates, all Nobel laureates of chemistry. Max Planck Institute funded me. So in the ship, I walked up to this person wearing that hat. He's none other than Linus Pauling, one of the most wonderful persons from the United States who advocated that you should eat vitamin C in large amounts to ward off cancer. So I walked up to him and said, "Sir, can I take a photograph?" This was when 1976, you can calculate how many years ago, 48 years ago. He said, no. I was taken aback. He said, no, I have to get my wife. So in the middle of the photograph is his wife. And we both are smiling. So also with me, with no longer gray hair at that time, but dark black hair. And on the ship in Lake Constance between Germany, Austria, and Switzerland. What did he say? He said, a genius who refuses to burn the midnight candle is a genius wasted. And he also said, I quoted in my book, the apparently silly questions the freshman chemistry students or freshman chemistry or other science students ask, the difference between me and other teachers is that I pursue those questions for 25 years or more, and invariably I come up with very new answers So there is nothing silly about questions, especially from young minds. So I think we have reached the stage today over just about an hour where the young people, including the faculty, if any, can ask me questions and we can handle them. There are no silly questions. Don't put yourself under pressure. Any question that you ask is very welcome. So I think I must conclude my talk with this photograph, celebrated photograph of mine with Professor Linus Pauling. Again in 2009, the Germans invited me. Again, I went to the Lindau Nobel Prize winners meeting. You know, Department of Science and Technology, Government of India selects about 60, 70 Indian students to go to Lindau every year to spend a large number of days or an entire week or more with Nobel laureates. On this note, let us close this lecture of mine and wait for any comments or questions. Thank you for your patience.
If you have any questions, please feel free to ask. So
Yes,
we have
sir.
a question.
पूछिए.
So yes,
Please.
there is question from Kirti Rani.
हाँ, क्या है question?
She
पढ़िए.
is suggesting that this is kind of out of topic, but I wanted to ask how to choose a title for research or review paper. What are the
हाँ, हाँ.
points we must look while choosing a topic?
बहुत अच्छा, बहुत अच्छा. Very, I think it needs a separate lecture on this question, but I'll try to give an answer in a few sentences. The choice of a topic of either review or experimental work is not such an easy one because it must be close to your heart. One has to be frank to oneself. मेरे को किस चीज में रुचि? मैं रसगुल्ला खाऊं, गुलाब जामुन खाऊं या समोसा खाऊं? या सबको जो मेरे सामने रखे हैं सब सारे गुलाब जामुन, सारे रसगुल्ले, सारे समोसे खा जाऊं? वो तो नहीं हो सकता ना. You have to choose what interests you. And you must know what is your strength. I know what is my strength. I can make new molecules. I can characterize them. I can ask somebody, please test them in a rat or a mouse or whatever against cancer. I can make some devices, nano devices. I can try and make measurements. तो आपको जो इंटरेस्ट है, पहले अपने इंटरेस्ट को पहचानिए. दूसरी बात, इस इंटरेस्ट को सपोर्ट करने के लिए बहुत पढ़ाई करनी पड़ेगी. आज तक पहले लोगों ने क्या काम किया है? We stand on the shoulders of giants of yesteryears. We stand on the shoulders of giants of yesteryears, and those giants also made mistakes. वहां हिम्मत से कहना चाहिए, अरे ये बड़े आदमी तो गलती कर दी, उनको समझ नहीं आई, मेरे को साफ नजर आ रहा है. ये हिम्मत होनी चाहिए. उसके बाद लगन होनी चाहिए. आपको काम करने के लिए इतनी हिम्मत आप में हो, 24 घंटे यदि काम करना है तो करने के लिए तैयार हैं. हम लोग, हमारा स्मॉल टीम दिन रात आपस में बातें करते हैं. मैं दिल्ली में नहीं हूँ, शायद 14 तारीख के आसपास मैं दिल्ली में रहूंगा, पर पक्का नहीं, अभी टिकट बुक नहीं हुआ, पर हम तब मिलेंगे दोबारा. पर ना मिले, आजकल मॉडर्न जो कम्युनिकेशंस इतने अच्छे हैं कि हम बिना मिले हुए भी इन पर्सन हम चीजें चर्चा कर सकते. पढ़ना बहुत जरूरी है. उसके बाद साइंस में एक्सपेरिमेंटल, इफ यू वांट टू डू एक्सपेरिमेंटल वर्क, यू शुड हैव प्लेस ऑफ वर्क. The place of work should have some reasonable facilities. You don't need sophisticated instruments. You can approach other institutes for help. As a young student, I do not know at what level are you. Can you tell me at what level are you? आप सेकेंड ईयर में हैं, एमएससी में हैं, किस में हैं? Undergraduate. So I told you, if you do not have facilities in your college, in your institutes, go to some very senior scientists in some of the well-known places in India and abroad. हमारे स्टूडेंट्स अब्रॉड भी समर इंस्टीट्यूट, समर इंटर्न के लिए जाते रहे बीच बीच में. तो आपको बड़े लोगों के साथ मुलाकात हो ही हो. आप जब आपकी मुलाकात होगी, उनसे आप प्रेरणा लेंगे, आप उनसे सीखेंगे, तो आप में ऊर्जा आएगी, आपको और हिम्मत आएगी. पर एक और बात है, What makes the mayor go? Money makes the mayor go. कुछ बहुत तो पैसा साइंस करने के लिए चाहिए, या तो गवर्नमेंट फंडिंग मिले. हमारे कोई 10 लड़के 2011 में 10 students, 2 other teachers from different departments took up a project of Delhi University. This is the rule, that there must be 10 students from different departments, 2 or 3 departments of the institutions, and there must be a kind of a mentor. So we had a mentor who was the director of solid state physics lab. Delhi University gave us 1 lakh. I'm not saying these young people produced technology, but over the year they interacted with me. They were not only students of chemistry, there were students of physics and so on, also a faculty from physics. So आपको कहीं ना कहीं किसी बड़े आदमी से सीखना पड़े. ये कहना मैं किसी से मदद नहीं लूं. आप जानते हैं क्या होता है, छोटा बच्चा घर में, बड़ी बहन कहते हैं मैंने किसी से नहीं सीखा, मैं तो अपने आप सीखा. ये सच्चाई नहीं है. तो इनवेरिएबली वी लर्न फ्रॉम ईच अदर. घर में बड़े लोग हैं, उनसे हम सीखते हैं. इसी तरह साइंस में हो, आर्ट्स में हो, डांस में हो, म्यूजिक में हो, थिएटर में हो, किसी बड़े नामी आदमी जिन्होंने अपने उपलब्धियां अपने लाइफ में करी हैं, पाई हैं, जिन्होंने नाम कमाया है, उनके साथ जाके पैर पकड़. डू यू अंडरस्टैंड माय हिंदी और आई शुड स्पीक मोस्टली इन इंग्लिश? आई हैव सेड मेनी थिंग्स. इट इज नॉट वन, दे आर ऑल कनेक्टेड टू ईच अदर. You should have interest, and this should exactly match your interest, not somebody else's interest. In India, very often students from say 10th class or even earlier are under tremendous pressure from the parents and the society. 12वीं क्लास का रिजल्ट जिस दिन आया, आपने वो कहानी पढ़ रखी है चेतन भगत की. पड़ोस की आंटी जो आपसे कभी नहीं बोलती थी, आके पूछती है भाई कितने नंबर आए, पास हुआ, फेल हुआ. तो उस कहानी में चेतन भगत ने कहा कि 92% लिए थे बेटे ने. बैंक मैनेजर थे, बड़े आदमी थे, पूरे घर में मातम छाया हुआ. क्या मातम? They were all sad that he won't get admission in any major institute or college. उसी टाइम पे लड्डू का बॉक्स लेके आई काम करने वाली. क्या बोलती है? साहब मेरा बेटा पास हो गया है, 43% है उसके मार्क्स, अंग्रेजी में भी पास हो गया, इज पास इवन इन इंग्लिश. दे आर सेलिब्रेटिंग, whereas here 92.5% they are in sad state of mind. We must be satisfied with what we have, but we must be all the time trying to do things, even if the situations and the conditions are not very friendly. Thank you for the question.
There is another question from Rohan
Yeah.
Kumar. He is
Yeah.
asking, from what I understand, the process of cancer drug production involves the discovery of a new variation of cancer in an organism. Then the drug production begins. However, why are variable types of cancer and their possible cure not pre-made in the labs before their actual discovery in the world?
Very difficult question. क्योंकि इसको जवाब कैसे थे? वो कह रहे नए नए varieties of cancer आ रहे. There are newer varieties. Why can't you anticipate this? Tell me, I knew the word endemic. I did not know the word pandemic. It was possibly there in 1811 or 1911, 100 years ago, 1911, Spanish flu. So, but again it came back in the 2020 era, and so many people died. He's asking the question, why did you not anticipate COVID-19? It happened 100 years ago. Why can't you make drugs well in advance? Well, in defense strategy, it's a very good point which he's making. You must have such intelligence, whether your neighbors who are inimical to you are actually going to attack you. In 1962, you know what happened. I was at that time still in school. Our then Prime Minister went and hugged the Chinese. Hindi Chini bhai bhai. They stabbed us in the back. We were underprepared. We had no rifles. We had only 1857 model rifles with which I did training for 3 years during my undergraduate days. Again, our other neighbor attacked us. We had to be prepared. Sometimes we are unprepared. We did not have intelligence on what is happening on the other side of the border. We cannot say, this is what the question is asking. Why can't we anticipate the newer forms of cancer? What mutations may happen? What changes may happen which may cause cancer? Can you not develop new drugs? Yes. In the case of COVID-19, you know how important the predictions of computational people was. Our student of physics, now with Ashoka University, he was on the TV, NDTV every day, every night, where they were predicting if this started in the month of, say, May, when will this actually end? And their predictions were quite close to the truth. It was no medical device, it's a computational study. They were very close to the date when this phase will end. Your question is very good. It's very difficult for anybody to anticipate types of cancer which have not been known. And therefore, since you do not know what the type of cancer will be, where is the question of developing drugs for varieties of cancer not yet known? The question is, maybe. I don't say cancer is a lifestyle disease. Diabetes, blood pressure, and so on are lifestyle diseases. But cancer may be an environmental disease in the sense that caused by poor pollution, bad conditions, and all kinds of viruses in the neighborhood of a person. So it's a very complicated question you are asking. How do we anticipate all these factors? Therefore, live in a good atmosphere, eat good fresh food, have antioxidants, make sure that any unwanted free radicals generated in your body are destroyed, even at the very beginning. So people have, there is another therapy in India which is very popular. There are many other therapies, I can't mention all of them. One of them is called naturopathy. Live in a more natural style. So your lifestyle, don't go by a car where the temperature is 80 degrees Celsius in summer. Now people have ACs, but when you enter a car, it's still very hot for the first few minutes. Some people put on the AC before they come into the car because it will not upset their body. But how much of luxury is needed? What I'm hinting is that in cancer, there could be environmental factors which are playing a role, which could be corrected if our environment is clean. The viruses are not allowed to proliferate. Even parasites from mosquitoes being transmitted to the human beings, those should be culled, they should be finished. So many other diseases that you will read about, that is that H1N1 virus that is often seen in Kerala, diseases in animals, in poultry, and so on. Thank you for your question. It's a very difficult question to answer. Roshan Kumar, can we read your question or hear your question?
Yes. So he is asking, has your research ever been disproved? How did you cope with it?
Very good question. A very loaded question. You see, what I learned in Germany in particular was that if you determined a certain value, a certain constant, say melting point, nobody in the world should get up and say that your melting point recorded in the lab was wrong. This is the greatest insult a scientist can suffer, that your observations are criticized and shown to be wrong. Today, what is happening, this young man must know that there is a lot of plagiarism, copy pasting. Even ChatGPT is helping in this process. So in many American universities and schools, ChatGPT is not allowed if a young person is to write. So whenever we submit any paper or a PhD thesis or a book, we have to test it with modern plagiarism software. Sujit, kitna percentage allowed hai, batana bhai?
Generally for research paper, under 10.
Under 10. And for others?
Thesis may be 10 to 12%. And some even journals in ACS and very high standard journal is less than 8% is acceptable.
What was the plagiarism value in your thesis certificate that you attached?
9. approximate 8.73%.
Now, you see, that question of yours is answered. Yes, there was one case I remember where we said that up to 150 degrees, this reaction did not occur. Somebody later on, because we didn't have heating facilities beyond that, we didn't have the solvents for that. So somebody did it at 200 degrees. He said, I'm able to do the reaction. This is not disproving my data. Up to 150 degrees, the reaction did not happen. At 200 degrees, we did not have the facility to heat it, so we did not observe. It cannot be wrong, said to be a wrong data. So your observations should be above any doubt, any questioning. Not today, not tomorrow. Even 100 years later, the result should not be questioned. Good question, but very difficult. But now rules are very strict. Even then, many papers from Nobel laureates are being retracted. You are reading this every day, and many people are being punished. So fortunately, I hope we have told the truth alone, and this will be proved not today, not tomorrow, but even 100 years later, that your work is not being questioned. Good question.
Thank you everyone for joining us on the 6th day of our lecture series. We will be releasing the feedback form shortly. Please take a moment to provide your valuable insights. Once again, I would like to thank Mr. Dr. Professor Eswaran for his informative and engaging lecture today. Thank you.
Thank you so much to all, one and all. Thank you. Bye.